So good afternoon, everyone. We will start our webinar today. So welcome and thank you for joining us today. So as usual, before we start, I would like to go through some housekeeping items. So during the webinar, please use the Q&A function if you have any questions to submit your question or vote for the existing ones. We will address them at the end of the session and prioritizing by the vote count and the order received. This webinar will be recorded, and the recording and slide deck from this session will be shared in a follow-up email, typically within 5 business days after the webinar. And the video recordings for this webinar and also the past webinars are all available at our YouTube channel. So if you are interested, just follow our YouTube channel. So before today's webinar, I would like to introduce this ICSA mentoring launch. The first launch was successfully launched last month, and we received very positive feedback, especially on the interactive discussion and some career suggestions and etc. So if you are interested, you can join the upcoming mentoring launch from statistics to technology industry. Yeah, they invited to data scientists from the Germany and also LinkedIn to discuss some questions. So one more thing, I would like to acknowledge the ICSA webinar committee for this series of webinar. This team works behind the scenes to identify speakers, organize logistics, and ensure we continue offering the events for our community. So for today, the webinar is Introduction to Multiplicity in Clinical Trials, which is an important topic. And we are excited to have an expert speaker with us today to guide us through this important topic. So now let's welcome our host, and Dr. Deli Wang, and our speaker, Dr. Yihan Li. I will stop sharing.
Okay, thank you, Sia. My name is Deli Wang. I'm a host of today's webinar for Dr. Yihan Li for the introduction of multiplicity in clinical trials. Before I hand over the floor to Dr. Li, let me introduce the speaker. Yihan is a director of biostatistics at Gilead Sciences. Currently, she is working on oncology clinical trials. Before she joined Gilead, Yihan spent the first 10 years of her career at AbbVie. So we was a colleague before. She working in immunology and rheumatology therapeutic area when she was in AbbVie. At AbbVie, she supported the drug development and the regulatory filings in multiple indications. She obtained her PhD in statistics from Pennsylvania State University. Without further ado, I'll hand over the floor to Yihan. Yihan, please.
Yeah, thank you, Deli, for the introduction, and thank you, Siyuan. Okay, so thanks ICSA for the opportunity for this webinar, and I'm happy to discuss the topic of multiplicity in clinical trials with you today. Here's just a disclaimer. So this webinar is aimed at providing an introduction and overview of multiplicity in clinical trials. I will provide a high-level review of the regulatory guidance on this topic, and we will walk through several commonly used multiple testing methods, including different types of gatekeeping procedures and the graphical approach. And we will use a hypothetical example to illustrate the use of the procedures. And at the end, we'll go over some general considerations if we have time. So multiplicity is present in virtually all clinical trials. Anytime you have multiple endpoints or multiple subpopulations of interest or multiple doses or otherwise multiple different treatment regimens compared to control, or you have multiple looks or interim analysis in a clinical trial, any of these would result in multiple hypotheses to be tested in the clinical trial. And in that case, you will have the issue of multiplicity. So if we don't properly handle or adjust for multiplicity, it will lead to an inflated rate of false positive conclusions. So in the context of clinical trials and drug development, this may lead to unsubstantiated claims for the efficacy of a drug. As a simple example, if we conduct 5 independent tests, each conducted at one-sided alpha of 0.025, then the chances of at least one false positive from these 5 tests is about 12%. And if we conduct 10 independent tests, the chance of at least one false positive is inflated to 22%. So you can see that the false positive rate inflates very quickly with the increased number of hypotheses. So this is something that needs to be addressed. So multiplicity occurs in all phases of clinical trials throughout the drug development process. From a regulatory perspective, though, the main concern is regarding phase 3 studies, which is the confirmatory clinical trials, because these are the studies that are intended to demonstrate effectiveness supporting drug approval and also supporting the efficacy claims in the drug labeling. So from a regulatory standpoint, we cannot tolerate an inflated possibility of false claims. Therefore, strong control of the study-wise type 1 error rate is typically required by regulatory agencies for confirmatory trials. Here, just to define some terminology, so type 1 error rate is the probability of incorrectly rejecting a true null hypothesis. So a true null hypothesis means that the truth is there's no treatment effect. So incorrectly rejecting it means we have a false positive result. So type 1 error is basically the chance of a false positive. Now the terms study-wise type 1 error, overall type 1 error, and family-wise type 1 error are synonymous terms. They all mean that the overall chance of at least one false positive test among all the hypotheses tested in the study. Also, you may notice that the regulatory usually requires strong control of the family-wise error rate. So what do we mean by strong control? So strong control means control of the probability of rejecting at least one true null hypothesis, regardless of which subsets of null hypothesis happens to be true. So basically, we need to control the chance of at least one false positive, no matter what the truth is for each hypothesis. So no matter which endpoints truly have an effect or not, we have to control the family-wise error rate under all scenarios. So let's take a high-level look at the regulatory guidance on this topic. So both FDA and EMA have issued guidance on the topic of multiplicity. The current FDA guidance was finalized in 2022, and the latest draft EMA guidance was issued in 2016. If you have not looked at these guidance before, I would definitely recommend you take a look and read through them. They are publicly available. And throughout the rest of the slides, all the text that is in dark green color are copied verbatim from one of the guidances. So in terms of the FDA guidance, I wanted to point out a couple things in terms of the scope of the guidance. So the FDA guidance is mainly focused on multiplicity issues in terms of multiple endpoints. It's pretty extensive in terms of laying out the different types of multiple endpoints and the different considerations for the different types of multiple endpoints. But other sources of multiplicity, such as multiple doses or time points or populations, are not specifically addressed in the guidance. The guidance mentions that making a false positive conclusion about effectiveness is a major concern, and that the guidance is intended to clarify when and how multiplicity due to multiple endpoints should be managed to avoid reaching such false conclusions. So it's a pretty focused scope. The other thing I want to point out is that the FDA guidance has an appendix that provides an overview of several statistical methods for multiplicity control. While this is not an official endorsement of these methods, it does cover a lot of the commonly used methods, and it's good to see the agency's summary and interpretation of these methods. The EMA guidance is a bit more loosely organized. It's almost like a Q&A. It covers a broader range of practical topics, but it doesn't go into as much depth sometimes. So in the next few slides, I will go over just a few high-level learnings from these guidance documents. Again, it's not meant to cover both guidances in depth, so I would encourage you to read through the documents yourself. So the first thing that should be emphasized that is covered in both guidance documents is prospective planning. So of course, all aspects of analysis of a clinical trial should be prospectively planned, especially for confirmatory trials. And in general, the statistical analysis plan should not be changed after unmasking of treatment assignments and performing statistical analysis. Here are some quotes from the two documents regarding prospective planning for multiplicity. So to control the type 1 error rate, it is critical that sponsors prospectively specify the following: all endpoints in the primary and secondary families, including all planned endpoints, time points, analysis populations, and doses, all data analysis that will be performed to test hypotheses about the pre-specified endpoints, regardless of whether they are considered primary or secondary. The analysis plan should describe the testing procedure for the hypotheses being tested with a proper control of overall type 1 error rate. In general, analyses of supporting or exploratory nature do not require multiplicity adjustment. Um, but anytime there is intention for the analysis to establish efficacy or a specific claim, or there's a chance to select a favorable result from many analyses, basically cherry picking, whether it's cherry picking for multiple endpoints, multiple populations, or even multiple analysis methods, in that case, pre-planned multiplicity adjustment and a valid confirmatory analysis strategy is needed. Next, both guidances discuss the typical hierarchy of families of endpoints. So the FDA guidance points out that endpoints in adequate and well-controlled drug trials are usually grouped hierarchically, often according to their clinical importance, but also taking into consideration anticipated drug effects. The critical determination for grouping endpoints is whether they are intended to establish effectiveness to support approval or intended to demonstrate additional meaningful effects. Endpoints that are critical to establish effectiveness for approval are often designated as primary endpoints. Secondary endpoints can provide useful description to support the primary endpoints or demonstrate additional clinical important benefits. The third category in the hierarchy includes all other endpoints, which are referred to as exploratory. Exploratory endpoints can include endpoints for research purposes or for new hypothesis generation. Each category in the hierarchy can contain a single endpoint or a family of endpoints. And the FDA guidance further points out that the overall type 1 error rate should control for the primary and secondary endpoint families altogether. When an effect on the primary endpoint is shown, that's when the secondary endpoints can be formally tested. In general, it may be desirable to limit the number of secondary endpoints. And exploratory endpoints do not need multiplicity adjustment because they are generally not used to support conclusions. Next, we'll talk about the types of multiple endpoints. So the FDA guidance discussed this in detail, mainly in the context of multiple primary endpoints. We will discuss 4 types of multiple endpoints here that are all listed in the FDA guidance. So the first type is the so-called co-primary endpoints. This is when we need to demonstrate treatment effects on 2 or more distinct endpoints in order to establish clinical benefit. In other words, determination of effectiveness depends on the success of all primary endpoints. So in this case, there's only one path that leads to a successful outcome. Basically, all primary endpoints need to be successful. So in this case, there is no opportunity for cherry picking. So there's no concern for type 1 error rate inflation and no multiplicity adjustment is necessary. However, something to keep in mind is that co-primary endpoints may increase the type 2 error rate, or in other words, power loss, because the bar is higher. The second type of multiple primary endpoints is when it's sufficient to demonstrate treatment effect on at least one of several primary endpoints. In other words, the success of any one of the endpoints can establish a drug's efficacy. In this case, because there are multiple chances of success, it will lead to type 1 error inflation and potentially a false conclusion that the drug is effective if we do not have multiplicity adjustment. So in this case, multiplicity adjustment is critical to control the type 1 error rate for the primary endpoint family. The other two types of multiple endpoints discussed in FDA guidance are composite endpoints and multi-component endpoints. For both types, the primary endpoint relies on multiple components, but ultimately a single variable is defined as a primary endpoint. So composite endpoint is often used for time to event outcomes, where multiple outcome events are of interest and importance. So a typical example is cardiovascular trials, where the primary endpoint can be a composite endpoint of major adverse outcome events. For example, a composite of myocardial infarction, stroke, or death. For multi-component endpoints, the endpoint variable is derived for each subject from multiple components using predefined rules or formulas. An example would be like in many rheumatic diseases, the primary endpoint can be based on a disease activity score that is a single score calculated from several clinical assessments or lab values for each subject. So for both composite endpoint and multi-component endpoint, as long as a single statistical test is performed on the overall defined endpoint, there is no multiplicity problem occurring for this endpoint. But the FDA guidance points out that it's also necessary to examine the individual components of the composite or multi-component endpoint. And depending on the purpose of examining the individual components, multiplicity adjustment may or may not be needed. So if the intent is to better understand each component's effect on the overall endpoint, then no adjustment is needed. However, if the intent is to establish additional effects of the drug on the components, then multiplicity adjustment should be made. Okay, so in the beginning, we mentioned there are many different sources of multiplicity. As mentioned in the FDA guidance, methods that apply to multiple endpoints also generally apply to other sources of multiplicity, such as multiple doses or multiple populations, etc. However, multiplicity due to interim analyses often has its own unique characteristics and challenges. Technically, you could try to apply a generic multiple testing method to multiplicity due to interim analysis, but that is generally not an efficient or preferred way. So neither the FDA or the EMA guidance actually directly address this topic, which is not to say that this is not important. It's actually a very important topic, but it's probably just too complex to be addressed in just one guidance. It probably deserves its own guidance, to be honest. The EMA guidance did refer these issues to the CHMP paper regarding adaptive designs. So we will not be covering this topic in this webinar either, just due to time limitations, but I wanted to point this out just because of the importance of addressing multiplicity issues in any clinical trials that have an interim analysis or have any adaptive design component. Okay, so next for the majority of the remaining presentation, we will be going over several commonly used multiple testing methods for clinical trials. There are, of course, many available statistical methods for multiple testing, and there are many ways to categorize the different methods. The one way that I like to group them is into non-hierarchical procedures versus hierarchical procedures, which is based on the structure of the endpoints that the procedures are suitable for. So examples of non-hierarchical procedures are Bonferroni, Home, Hochberg, or Dunnett test. Of course, there are others. I'm just listing a few examples. These tests are used when you have a group of endpoints that don't have a clear hierarchical structure or relationship among them. And you're just trying to reject as many null hypotheses as you can without really caring about the order of which one gets tested first. On the other hand, hierarchical procedures is often used when there is distinct order or hierarchy or specific relationship among the endpoints that you want to take into account in the testing. Examples include fixed sequence test, fallback procedure, gatekeeping strategies, and graphical approach. Again, this is not an exhaustive list. So the methods in bold here are the ones that we will be discussing in detail today. And these methods are also all included in the FDA guidance. So first, let's go over a few classic non-hierarchical methods. So Bonferroni is probably one of the oldest and most classic multiple testing methods, and you're probably familiar with it. Basically, we divide the available total alpha equally among the multiple endpoints. So if there are m endpoints, then the critical value for each endpoint is alpha over m. And any endpoint that has a p-value less than alpha over m is considered significant. A natural extension of the original Bonferroni is the weighted Bonferroni, where instead of using equally divided weights among hypotheses, we can split the alpha with different weights, as long as the sum of the weights is equal to 1. As mentioned in the FDA guidance, these weights should be pre-specified in the design of the trial, taking into consideration the clinical importance of the endpoints, as well as the likelihood of success or other factors. So the biggest advantage of the Bonferroni method is very easy to use and simple to explain to non-statisticians. It is a single-step test, which means the critical values used for testing each hypothesis is completely predefined and remain constant. The method is completely assumption-free, so strong control of the family-wise error rate is guaranteed by the Bonferroni inequality. The FDA guidance mentions that a Bonferroni can be applied without concern for the endpoint types, without concern of the statistical distribution or the type of correlation structure. Of course, the simplicity of this method comes at a cost. So basically, the test is conservative, meaning it's not always taking advantage of the full alpha. The actual family-wise error rate is often less than the full alpha, which means there is room to improve the power. So in confirmatory clinical trials where we want to usually maximize the power as much as possible, we actually do not often use the original Bonferroni method, and we would typically try to use a more powerful method. So that brings us to the next method, the home procedure. The home procedure is a multi-step, step-down procedure, and here are the steps to perform it. So first you order all the endpoint p-values from smallest to largest, p1 through pm. You compare the smallest p-value, p1, to alpha over m. If p1 is less than alpha over m, the endpoint for p1 is considered significant. And next we compare the second smallest p-value, p2, to alpha over m minus 1. If p2 is less than alpha over m minus 1, the endpoint for p2 is considered significant. And next we'll compare the third smallest p-value, p3, to alpha over m minus 2, and so on. So the procedure continues as long as pi is less than alpha over m minus I plus 1, all the way until pm, which is compared to alpha. However, if at some point we have a pi that is greater than or equal to alpha over m minus I plus 1, then the procedure stops. And the remaining larger p-values are not evaluated and cannot be considered significant. And there is also a more general weighted version of Holmes, which allows unequal alpha allocation to the individual null hypothesis. So the testing follows a similar pattern, but the critical values may be adjusted a little bit. However, the critical values are getting bigger and bigger as you keep on testing. So the home procedure is uniformly more powerful than the Bonferroni method. Um, so as you remember, for Bonferroni, all the endpoints are tested at alpha over m. But for home, the smallest p-value is tested at alpha over m, but at each subsequent step, the remaining endpoints get tested at increasingly larger critical values. So Holmes will always end up with more significant endpoints than Bonferroni. At the same time, Holmes can also guarantee strong control of the family-wise error rate without any assumptions, which is the same as Bonferroni. So in reality, there's really no reason not to use Holmes over Bonferroni. It is more powerful and it guarantees control um, it, without any assumptions, just like Bonferroni. If we have to list the disadvantage of Holmes, we can say that we cannot fully predetermine the critical values for each endpoint, and we cannot fully determine the testing order because it depends on the test results. However, that's not really a disadvantage. It's just the nature of any stepwise testing procedure. And usually the stepwise testing procedures will be more powerful than a single step procedure. Okay, next we move on to the Hochberg procedure. So both Holm and Hochberg are stepwise procedures. and they're actually use the same set of critical values. So it's sometimes easy to mix up the two. So Holmes is a step-down procedure, whereas Hochberg is a step-up procedure. So what do we mean by step-down or step-up? So it refers to the order of testing. So for both procedures, we order the p-values from smallest to largest. Holmes starts testing from the top. So we start testing from the smallest p-value and we go down the list. Whereas Hochberg starts testing from the largest p-value, which is at the bottom of the list, and it goes up. So for Hochberg, as we mentioned, we start by comparing the largest p-value to alpha. And if the largest p-value is less than alpha, then all endpoints are considered significant, and we do not even need to test the remaining endpoints. Everything will be considered significant. If the largest p-value is not less than alpha, then we look at the second to largest p-value, pm minus 1, and we compare that to alpha over 2. If pm minus 1 is less than alpha over 2, then the endpoint corresponding to pm minus 1 and all the remaining endpoints with smaller p-values are considered significant. If that is not true, then we move on to the next largest p-value and so on. So the procedure continues until we find a p-value that is smaller than its corresponding critical value for the first time, and then the procedure stops at that point, and that endpoint and all the remaining endpoints with smaller p-values are considered significant. So here's a slide that we have a side-by-side illustration of Holmes step-down procedure versus Hochberg step-up procedure using an example. So suppose we have 5 hypotheses and we order them based on the test results from the smallest p-value to the largest p-value. And we also calculate the critical values for each hypothesis, which is based on the order of the hypothesis. So the smallest p-value is compared to alpha over 5. So suppose the total offer is 0.05, then the critical value for the smallest p-value is 0.01. And the next one is compared to alpha over 4, and so on and so forth. And the largest p-value is compared to alpha, which is 0.05. And again, the critical values are the same for both Holm and Hochberg. It's just the order of testing that is different. So now for Holmes step-down procedure, we start testing from the smallest p-value and we step down. So in this example, the first p-value is smaller than the critical value, so we can keep testing. The second p-value is also smaller than the critical value, so we can keep testing. The third value is bigger than the third p-value is bigger than its critical value. So here we will stop testing and we can no longer test the remaining hypotheses. So for Holmes procedure, we can claim the first 2 p-values are significant. Now for Hochberg step-up procedure, we start testing from the largest p-value and step up. And for Hochberg, remember that we keep testing until we find the first p-value that is smaller than the critical value. So we start from the bottom. The largest p-value is not smaller than the critical value, so we keep testing. The next largest p-value is smaller than the critical value, so we can stop testing. And we no longer need to test the remaining hypothesis. And per the Hochberg procedure, we can claim the fourth hypothesis as well as all the hypotheses that have smaller p-values to be significant. So from this illustration, you can see how Holmes steps down and how Hochberg steps up. And we can also see that Hochberg ends up claiming more hypotheses being significant. And that is not just true for this example. Actually, Hochberg is uniformly more powerful than Holm, meaning if an endpoint is significant under Holm procedure, then it will also definitely be significant under the Hochberg procedure, but not necessarily vice versa. However, and this is a big caveat, Hochberg is not assumption-free. So as mentioned in the FDA guidance, Hochberg procedure is known to provide adequate overall alpha control for independent endpoint tests or for positively correlated dependent tests with standard test statistics in some cases. For example, the test statistics are jointly bivariate normal. Various simulation experiments for the general case, for example, for more than 2 endpoints with unequal correlation structures, indicate that the Hochberg procedure usually will, but is not guaranteed to control the overall type 1 error rate for positively correlated endpoints, and it fails to do so for some negatively correlated tests. So that is a big caveat for Hochberg procedure, which is unlike Holmes, because Holmes can guarantee strong control of the family-wise error rate without any assumptions, whereas Hochberg does require some assumptions for the family-wise error rate to be maintained. In reality, the assumptions required for Hochberg procedure may be hard to fully verify, but Hochberg is still used in practice a lot because most of the times we assume that there is some positive correlation structure across the endpoints that we're testing. However, if you want to use Hochberg for multiplicity adjustment in a confirmatory trial, you should consider upfront communication and discussion with a regulatory agency. Next, we'll move on to the hierarchical methods, and we'll start from the simplest one, the fixed sequence test. So in many studies, testing of the endpoints can be ordered in a specified sequence, starting with the primary endpoint and followed by the secondary endpoints, often ranking them by clinical relevance or likelihood of success. I'm sure many of you are familiar with the fixed sequence test. Basically, we predefine the order of the endpoints and we use the same significance level alpha to test each endpoint, but only moving to the next endpoint in the sequence after the success of the previous endpoint. So as shown in this diagram, the full alpha is passed on from one endpoint to the next in the pre-specified order. So the key points of the fixed sequence method is one, prospective specification of the testing sequence, and two, no further testing can be done as soon as one endpoint in the sequence fails to show significance at level alpha. So the fixed sequence test is very easy to use and explain, and it does not require any adjustment of alpha for the individual test. Each endpoint is tested at the full alpha as long as the testing sequence doesn't break. So the disadvantages are also pretty obvious. If one endpoint in the testing sequence cannot achieve statistical significance, then all remaining endpoints in the chain cannot be tested, no matter how small their p-values are. So there is some power loss expected for endpoints that are ranked lower. Therefore, as pointed out in the EMA guidance, careful selection of the order of the test of the hypothesis is critical. The guidance also points out that, in principle, the planning and assessment of a clinical trial should prioritize those endpoints of greatest interest from a clinical perspective. But it has become common practice to rank endpoints based on the likelihood that the individual null hypothesis can be rejected. Ideally, the clinical assessment should focus on those endpoints of greater clinical importance. Next, we will spend some time discussing a class of multiple testing methods called gatekeeping procedures. Gatekeeping basically means you have to pass a test for some endpoints first, and they act as a gate before you can open the gate and move on to test other endpoints. So the fixed sequence test that we just talked about is basically a very simplified case of gatekeeping, where each endpoint is a gatekeeper for all the subsequent endpoints. So formally speaking, gatekeeping procedures was proposed by Dimitrienko et al.'s paper in 2008. It addresses problems of testing hierarchically ordered families of hypotheses. So families usually correspond to primary and secondary objectives in a clinical trial, and inferences in each family depend on the acceptance or rejection of null hypothesis in the earlier families. So that's a very high-level description of gatekeeping that was copied from the FDA guidance. Of course, there can be more than two families of endpoints defined if needed, and in each family, there can be one or two or more endpoints. So exactly how the testing in each family works and how the alpha is passed from one family to the next is very much dependent on the specific gatekeeping procedure. So there's many different types of gatekeeping procedures. In the next few slides, we will discuss two basic types of gatekeeping procedures, namely serial gatekeeping and parallel gatekeeping. So for serial gatekeeping, we start with testing endpoints in family 1 at the overall alpha level. If there are multiple endpoints in family 1, then we need to apply a multiple testing method within family 1 so that the family-wise error rate within family 1 is controlled at alpha. If all endpoints in family 1 are significant, we can pass the overall alpha to family 2, and now family 2 can be tested at the overall alpha level. However, if any endpoint in family 1 fails to be significant, then family 2 cannot be tested at all, and the overall testing stops. So the advantage of serial gatekeeping is that it strictly preserves the hierarchical order structure of the endpoint families. So family 1 is the gatekeeper for family 2, and you have to pass all the endpoints in family 1 in order to move on to family 2. The other advantage is that within a given family, endpoints can be tested at the overall alpha level using any pre-specified acceptable method that controls the type 1 error rate within the family. So basically, you can use whatever multiple testing method you want within the family. It can be Bonferroni, Home, Hochberg, fixed sequence, or any complicated method you want, as long as it controls the family-wise error rate within the family. There's no restrictions on what to use. The disadvantage of serial gatekeeping is similar to fixed sequence tests. So even if only one endpoint in family 1 fails to be significant, then family 2 cannot be tested at all. Now let's look at another type of gatekeeping called parallel gatekeeping. So again, we start with testing endpoints in family 1 at the overall alpha level, but this time we cannot use whatever method we want within family 1. We can only use what's so-called a separable testing method, and we'll talk about that a little bit more in a minute. So in the original parallel gatekeeping method, Bonferroni is used for testing within family 1. So now the good news is we do not have to rely on all endpoints in family 1 to be significant to test family 2. So as long as one endpoint in family 1 is significant, we are able to pass some alpha to family 2 and test family 2. Exactly how much alpha can be passed to family 2 depends on how many endpoints or which endpoints are significant in family 1. If all endpoints in family 1 are not significant, then testing stops. So now let's take a step back to explain what does it mean to be a separable procedure. The definition of a separable procedure is one that does not exhaust all the alpha unless all null hypotheses are true. So Bonferroni is a separable procedure. However, Home and Hochberg are not separable procedures. So a simplified explanation is that Home or Hochberg maximizes the use of alpha within the family, so there's nothing left to pass to the next family, unless all the endpoints in the first family are significant. So that's why they're not separable. Um, a separable procedure like Bonferroni is usually less powerful, meaning they leave some alpha on the table unused, which is what allows it to pass some alpha to the next family, even if not all endpoints in the first family are significant. So there are methods that are more powerful than Bonferroni, but are still separable, such as what's called the truncated home procedure is an example, which is basically a kind of a middle ground between a Bonferroni and a home procedure. Okay, so how much alpha is passed from family 1 to family 2? If you remember with serial gatekeeping, when alpha is passed from family 1 to family 2, it is all or nothing. So either the full alpha is passed to family 2 or family 2 cannot be tested at all. But with parallel gatekeeping, the amount of alpha passed to family 2, it can vary. So it depends on what multiple testing procedure is used for family 1 and which endpoints were significant in family 1. If all the endpoints in family 1 are significant, then the full alpha can be passed to family 2, regardless of what method was used in family 1. That is the same as serial gatekeeping. But if not all endpoints in family 1 are significant, then it depends on what method is used and which endpoints were significant to determine how much alpha is passed to family 2. So just as an example, if we have two endpoints in family 1 and only one of them is significant, if we used the Bonferroni method for family 1, then half of the alpha can be passed to family 2, which is basically the amount of alpha corresponding to the endpoint that was significant. If we use truncated home for family 1, then only a portion of the half of alpha can be passed to family 2. And the portion depends on the truncation factor gamma, which is the parameter defined in the truncated home procedure. And gamma is between 0 and 1. So we're not going to have time to go into detail of truncated home, but just to explain two extreme scenarios. When gamma is equal to 0, truncated home is basically equivalent to Bonferroni. As you can see here, in that case, you're passing alpha over 2 to family 2, the same as Bonferroni. And gamma has to be between 0 and 1, but gamma cannot be 1, because if gamma is 1, then you can see that there's no alpha passed to family 2. In that case, it is not truncated home anymore. It is basically just homes. And so that's no longer a separable procedure. In that case, it's basically not parallel gatekeeping anymore, and you're just going back to serial gatekeeping. Okay, so compared to serial gatekeeping, parallel gatekeeping has more opportunity to pass alpha to the next family. It does not require all endpoints in the previous family to be significant to be able to pass some alpha to the next family. That's why in the illustration of parallel gatekeeping, we have arrows pointing from each endpoint to the next family, because the success of each endpoint allows a pathway to pass some alpha to the next family. Whereas if you remember for serial gatekeeping, we had one combined arrow pointing from family 1 to family 2, because it requires the entire family 1 to be significant in order to pass all of the alpha to family 2. Now, when all the endpoints in family 1 are significant, then all alpha can be passed to family 2, no matter whether it's serial or parallel gatekeeping. So for parallel gatekeeping, there's more opportunities to pass alpha to family 2, but it comes at a cost that family 1 can only be tested using a separable procedure, which is less flexible compared to serial gatekeeping, because serial gatekeeping, you can use whatever procedure you want for family 1. So you can basically pick a more powerful procedure for serial gatekeeping for testing within family 1, whereas for parallel gatekeeping, you're restricted to using separable procedures, which are can be less powerful. So it's basically a trade-off of having more chance to pass alpha to family 2 versus preserving more power for testing within family 1. There's another less obvious disadvantage of parallel gatekeeping, is that the hierarchical structure of the endpoints is not always strictly preserved. And we can use a hypothetical scenario to illustrate this point. So suppose we use Bonferroni to test within family 1, and the primary endpoint for dose 1 is significant, but the primary endpoint for dose 2 is not significant. And based on parallel gatekeeping, we can pass half the alpha to family 2. Note that the alpha is passed to the whole secondary endpoint family and not passed to a specific endpoint within the family. So now we have alpha over 2 used to test the whole of family 2. Suppose that both secondary endpoints are, have very small p-values, so both of them are significant under alpha over 2. So now we end up with this scenario where the primary endpoint of dose 2 is not significant, but the secondary endpoint of dose 2 is significant, which is kind of a illogical scenario. And this is possible under parallel gatekeeping, but it's not possible under serial gatekeeping. Of course, on the flip side, we can use the same example to show that that the advantage is with parallel gatekeeping, because if we had used a serial gatekeeping, um, if we fail to claim those two primary endpoint to be significant, then we cannot test the secondary family at all, right? So we will not be able to claim the secondary endpoints. So I guess the point is there is no uniformly better choice between serial gatekeeping and parallel gatekeeping. It depends on the specific study scenario and what is prioritized. In addition to serial and parallel gatekeeping, the FDA guidance also mentions multi-branched gatekeeping. So as mentioned in the guidance, some multiplicity problems are multidimensional. For example, a trial could have multiple endpoints and multiple dose groups that each will have those endpoints tested. And on top of that, there could even be multiple hypotheses regarding each endpoint. For example, we may conduct both non-inferiority and superiority tests on each endpoint for each dose. So the multiple sources of multiplicity create the potential for multiple pathways of testing the hypothesis. And multi-branch gatekeeping procedure allows for ordering the sequence of testing with the option of testing more than one subsequent endpoint if a preceding test is successful. So here's an example from the FDA guidance. So we start by testing non-inferiority for the primary endpoint. If it is successful, then a portion of the alpha is used to test the same endpoint for superiority, while another portion of the alpha is used to test non-inferiority of the secondary endpoint. And if that one is successful, then the corresponding alpha is passed on to test the superiority of the secondary endpoint. So alpha is not passed along necessarily in a linear fashion, but passed on to multiple branches. So this is actually a really good segue for our next topic, which is the graphical approach. This is the last class of methods that we'll discuss today, and it's actually a personal favorite of mine. So the graphical approach is a stepwise multiple testing procedure proposed by Frank Bretz et al. In 2009. It uses graphs with nodes and directed weighted edges to represent multiple testing procedures, and it strongly controls the family-wise error rate without any assumptions. And the best part is that it is very intuitive. So we have already been using the concepts of the graphical approach throughout the presentation without formally defining it, such as these diagrams that we use to illustrate Bonferroni as well as the fixed sequence test. So while these are very simple examples, they actually perfectly showcase the two key elements of the graphical approach, which is alpha allocation and alpha propagation. So with Bonferroni, you can see that we are splitting the alpha and allocating a portion of the alpha to each hypothesis, which is alpha allocation. And with fixed sequence, we allocated all the alpha to the first hypothesis, but as we successfully reject a null hypothesis, we are propagating the alpha to the next hypothesis and depicting by these arrows here. So that shows alpha propagation. So the graphical approach basically incorporates these two concepts, alpha allocation and alpha propagation, into a formal framework that guarantees strong control of the family-wise error rate. So to formally define it, each hypothesis is represented as a node, and the overall significance level alpha is split as weights and assigned to each hypothesis. And alpha propagation is denoted through weighted directed edges, which define a transition matrix that determines how alpha is transferred between hypotheses. So there are some very basic and natural constraints that need to be satisfied. Namely, the sum of the alpha allocation to all the nodes should be no more than alpha, and the sum of the weights that come out of each node should be no more than 1. The testing follows an iterative process. At each step, the p-values for each hypothesis or each remaining hypothesis is compared with the allocated alpha. And if one of the null hypotheses is successfully rejected, then we follow the algorithm defined in the paper to update the graph for the next step, which means we will remove the node corresponding to the successful hypothesis and update the alpha allocation for all the remaining nodes and update the transition matrix as well for the remaining hypothesis. So here we show as an example what the initial graphs or what the initial alpha allocation vector and transition matrices look like for the Bonferroni and the home procedure. So here's the initial alpha allocation vector. So for both Bonferroni and home, the initial allocation is half an alpha to each hypothesis. But Bonferroni did not define any alpha propagation, so the transition matrix is all zeros, meaning there's no transfer of alpha between the hypotheses. For Holmes, the transition matrix is defined such that whichever null hypothesis is rejected first, the alpha for that hypothesis will be fully transferred to the remaining hypothesis. So for Holmes procedure, suppose we reject H1 first, then this is what it looks like to update the alpha allocation vector and update the transition matrix. So if we reject H1 first, then all the alpha from H1 is passed to H2. So now H2 has a full alpha. And because there's only one hypothesis remaining, there's no more transfer of the alpha, and the transition matrix is all zeros. So to summarize, the initial graph, which consists of the initial alpha allocation vector and the transition matrix, defines a unique testing procedure. And whenever a null hypothesis is rejected, the graph is updated based on the algorithm. So the key elements of a graphical procedure are the initial alpha allocation and the initial transition matrix. And then with the iterative algorithm, they uniquely define a sequentially rejective test procedure that strongly controls the family-wise error rate. Here are some advantages of the graphical approach. So first and foremost, any graphical procedure, no matter how simple or how complicated, as long as it's defined and carried out following the framework, is guaranteed to strongly control the family-wise error rate, and it does not rely on any assumptions. Actually, a lot of commonly used multiple testing methods are covered under the graphical approach. So we have seen a few examples like Bonferroni and Holmes, and also most of the gatekeeping procedures can be represented using the graphical approach as well. But what I think makes it really cool is how intuitive and easy it is to use the graphical approach to construct new customized procedures. And because the alpha allocation and the propagation is very explicit and flexible, we can easily use the graphical approach to tailor to different types of logical hierarchies or relationships among the endpoints. So disadvantages. The first one was it's not really a disadvantage. Again, the final critical value used for each endpoint and the testing order cannot be fully predetermined, but that is true of any stepwise procedure. The other disadvantage is that the graphical approach is a Bonferroni-based procedure. It is based on Bonferroni and the close testing principle. So because it's based on Bonferroni, it does have potential for power improvement. And there are extensions in the literature for the graphical approach that are based on multiple testing procedures outside of Bonferroni, and so they will usually come with a power improvement. Although I would say most of the times the power improvement is pretty small, and it does come at the cost of greatly increasing the complexity of the procedure. Okay, so now we'll go over an example to illustrate the use of the graphical approach as well as the gatekeeping strategies. So suppose we have a clinical trial with one primary endpoint and one secondary endpoint, and there are two doses of active treatment compared with placebo. So in total, we have 4 hypotheses. So logically, for a given dose, the secondary endpoint can only be tested if the corresponding primary endpoint null hypothesis is rejected. And between the two doses, there's no order of testing. So we want to be able to recycle the alpha between the two doses. So here are some different ways that we may design the graphical testing procedure. We start from a simple one. In this case, we allocate half the alpha to the primary endpoint of each dose. And then if the primary endpoint hypothesis is rejected, we pass the alpha to the corresponding secondary endpoint. But we can see here that once the alpha gets to the secondary endpoint, there are no arrows coming out of the secondary endpoints. And this is what I call a dead end. So that the alpha ends up here, but it's stuck here and it's not being reallocated anywhere else. So whenever you see a dead end, it means that there's potential for better reuse of the alpha that can potentially improve the power. So to that point, we can add these arrows that transfer the alpha from one dose to the other dose, so that if both the primary endpoint and secondary endpoint of one dose are significant, we can reallocate that alpha to the other dose, so that the other dose can now be tested at the full alpha. So this procedure is going to be uniformly more powerful than the first procedure. From here, there may not be any more uniform improvements that we can make, but we can explore various adjustments depending on the prioritization of the different endpoints. For example, if we want to increase the chances of claiming both primary endpoints significant, and we want to deprioritize the secondary endpoints, then we may consider something like this, where we add the transfer paths between the two primary endpoints, so that once the primary endpoint of one dose is significant, we can pass some of the alpha to the primary endpoint of the other dose and pass less alpha to the secondary endpoint. So now let's take this last procedure we created and use that as an example to walk through the testing process. So this is the initial graph that defines this procedure. And this is what the initial alpha allocation vector looks like and what the initial transition matrix looks like corresponding to this graph. And remember that we will iteratively update the alpha allocation and the transition matrix at every step throughout the testing. So suppose these are the p-values that we observe in this trial, and suppose the total alpha is one-sided 0.025. So at each step, we compare the p-values to the allocated alpha. So in the beginning, H1 and H2 each have half alpha assigned, which is 0.0125. So we can see that the p-value for H1 is less than the assigned threshold, so we can reject H1. And now we update the graph. We can remove H1 from the graph. And this is what the updated graph would look like. So first, we notice that the allocated alpha for the remaining hypotheses are all updated. So H2 originally had half an alpha, and now it has 3/4 of the alpha. And H3 originally had no alpha, and now it has 1/4 of an alpha. And in addition, the transition matrix is also updated. I think we're running a little bit low on time, so I'll try to go a little bit faster. But basically, the transition matrix tries to preserve the pathways between hypotheses, but the actual weights will be updated based on the algorithm. As you can see here, like this path was originally passing half of the alpha allocated, but now after update, it's updated to 2/3 of the alpha. So the next step, we compare, again, all the remaining p-values with the updated alpha allocation. And in this case, we can see that we can reject H2. So we remove H2 from the graph, and we update the graph again. And the next step, we compare the remaining p-values, and we can see that H3 p-value is smaller than the alpha allocated here, so we can reject H3. And this is the final graph with only one hypothesis left. And in this case, we're not able to reject H4. So these were the 3 graphical procedures that we designed for this example. And we walked through the testing of this third procedure, and we were able to reject or claim the significance of these 3 endpoints. And we could not claim significance for H4 based on these observed p-values. With these same observed p-values, if we did the testing using the first procedure, we would have been able to reject both endpoints for dose 1, but we would not have been able to claim significance for any endpoint for dose 2. If we had done the testing for using the second procedure, it would have had the same result as the third procedure in this particular case. And we can also try to apply the gatekeeping procedures to this example. On the left, we have serial gatekeeping, where both families are tested using Holmes. And on the right, we have parallel gatekeeping, where the first family is tested using Bonferroni, and the second family is tested using Holmes. And I'll try to go through this quickly, but basically, if we use serial gatekeeping, we would have been able to reject both primary endpoints and pass the full alpha to family 2, and we would have been able to reject H3, but not H4. And in this particular example, using these observed p-values, if we did parallel gatekeeping with Bonferroni in family 1, we would be able to reject or claim significance for H1, but not for H2. So we can pass the alpha from H1 down to the second family. And in this case, because we only were able to pass half the alpha to family 2, and with these observed p-values, we actually were not able to reject either of the secondary endpoints. Okay, so see if we can go through this last slide of consideration. So usually there's not one uniformly most powerful multiplicity control strategy. So some key factors to consider when deciding what strategy to use for a clinical trial include the importance or clinical relevance of the endpoints, other logical restrictions or relationships among the endpoints, the expected power of each endpoint, any correlations across endpoints, as well as the team's tolerance of the failure of different endpoints. So cross-functional input is very important. It can help direct the choice of multiplicity strategies. But from a stats perspective, we can always conduct simulations to inform the final selection of the testing procedure, as well as fine-tune the parameters of the procedure. And the simulation settings should ideally cover a range of different effect sizes and also consider different correlation scenarios. This is a slide saying that there are other types of multiple testing problems or methods that are not covered in here, such as the problems of multiple dose groups compared with a single control group. And as we mentioned, the problem of group sequential designs or adaptive designs where there's interim analyses. So I listed some possible methods to consider for these types of problems. So that was all, and here are some key references. And thank you for your attention. I'm not sure if there's any time for questions. I'm happy to stay longer.
Yeah, thank you very much, Yihan. I think this is a wonderful presentation and very detailed, from simple procedure to complex procedure. Yes, we do have one question in the QA. Maybe Yihan, you want to? Yeah, two questions right now.
Okay.
Oh yeah, just the first question is just maybe open a QA. I think the second question just ask the slides sharing. I think no problem, we'll share. The first question, maybe Yihan, do you want to read and also?
Oh, okay. Let me see. Okay, in fixed sequence procedure graphical approach, we may use, for example, 0.05 for both primary and secondary endpoints, so-called alpha allocation, alpha transfer. I know that the second test depends on the first test result significance, but why does the procedure not inflate the family-wise error rate? In comparison, when we consider group sequential design, we still depend on interim analyses to go, no go, but there's all alpha spending. Do you have any insight, mathematical justification, references to distinguish these two methods? Okay, so in the fixed sequence test, right? Let me see if I can go back, but I think we know what the fixed sequence test looks like, right? So the testing of the second endpoint depends on the success of the first endpoint. So it's conditional, right? So only if the first test, if the p-value of the first test is less than, let's say, 0.05, only then can you pass the full 0.05 to the next test, right? If the first test failed at 0.05, then the second test cannot be tested at all. I think in the case of group sequential, if you applied, you could apply that, but nobody would do that because you would not want to bet everything on the interim analysis, right? If you use the fixed sequence test in group sequential, that would mean that you would put all of your alpha on your interim analysis. And if your interim analysis is not successful, then you won't be able to test the final. But that's not the point of an interim analysis, right? So nobody would do that. So, so you would so you would allocate some of the alpha to the interim analysis and then the remaining for the final analysis. Now, again, you could use a very simple method like Bonferroni, right? Theoretically, you could use Bonferroni with a group sequential trial. You could say put 0.025 for the interim analysis and put the remaining 0.025 to the final analysis. If you did that, there's no problem at all from a type 1 error control perspective. But again, the reason you don't do that is because there's usually more powerful methods. So like the group sequential alpha spending functions allow you to put, alpha 1 for the interim and alpha 2 for the final, but alpha 1 and alpha 2 usually add up to more than the total alpha, right? And so that's why you would usually use something that's more tailored to the group sequential setting. But you could technically use things like Bonferroni or fixed sequence. They would work the same way. So alpha control is the same way, but you know, it's just not as powerful or as tailored to the group sequential scenario. I hope that answers the question.
Okay. Thank you very much, Yihan. I think that's the only question we received here. A second question, just slide sharing, we'll share. But thank you again, Yihan, and thank you everybody joining the webinar. I think let's close today's webinar. Have a good weekend.
Thank you very much, everyone.
Thank you.